I'm not getting audio. Hi, this is Pat Whitcomb. It's time, so we'll start the webinar. Today I'll be talking about how to deal with hard to change factors in a designed experiment, and the tool we use are split plots. And I'll talk about the advantages or the pros, and also some of the disadvantages or the cons. Since there's a lot of people listening, I'm going to have the attendees muted so that only I can speak. But if you have questions, you can click on the little question box and type those in. And Sherry Kraber is going to be sitting in answering your questions as I present the webinar. So why don't we just get started? First of all, I want to make sure that people are hearing me. So if you can click on the raise hand, that will tell me whether or not you can hear me. I'm not seeing any hands go up. So does that mean no one can hear me? Oh, there's some. I see them now. Okay, so I'm going to go ahead and start then. So I'm basically, my webinar today will have 4 parts. It'll take between 50 and 60 minutes. And I'm going to be talking about split plots and the fact that specifying hard to change factors, we put a restriction on the randomization. And then rather than going into a lot of technical detail, I'm going to illustrate it with 2 examples, one with a factorial split plot and one with an RSM type response surface design type, where we're going to be studying a combination of mixture and process factors in a split plot. Then I'll summarize. So what is a split plot and what is the restriction on the randomization we're talking about? Okay, so the problem in some designed experiments is that we have some factors, maybe temperature if we have a very large oven that takes a long time to equilibrate, or others that are difficult, time consuming, or expensive to vary. And in some cases, if we did a completely randomized experiment so that we were varying these things often, that experiment would become impractical or possibly impossible to do. So what's the solution? Well, we restrict the randomization. So maybe some of you, I won't point any fingers, but one of us, when I was much younger and we didn't have good statistical software, what I would do would be to sort on a factor that was hard to change and then reset the run order so that I didn't have to change that factor very often. That's basically what we do with a split plot. What's different is now with good software, we can take that randomization restriction into account when we do the analysis. And so that's what we're going to be doing with split plots. So that creates a split plot. So you can think about whether or not you have factors that are difficult to randomize, and then those would be the hard to change factors that we'd vary less often when we do a split plot. So where does the name split plot come from? Well, split plots, as much of designed experiments, were originally developed in the field of agriculture. And so what would happen is experimenters would want to apply a treatment. Let's say that our treatment that we want to look at is a herbicide. And we want to apply that and see what effect that herbicide has on the yields of different crops. So in this little example, I've got 3 different herbicides, A, B, and C. And then I want to know the effect of those herbicides on the yields of corn, potatoes, and squash. Now, if we did a completely randomized experiment, we have little plots of corn and squash and potatoes, all randomized over a large area. And then we'd go in and to each one of these little plots, we'd try and apply a herbicide. Now, if we're spraying that herbicide, you're going to have a problem in that you're going to get drift. And so if I tried to put a herbicide on the corn and right next to it was the potatoes, which was going to get a different herbicide from the corn could drift onto the potatoes, and the herbicide from the potatoes could drift onto the corn, and you wouldn't be able to separate the effects of the individual herbicides. So what you might want to do is say, okay, I'm going to do a bigger plot where I'm going to put herbicide B, and then I'm going to divide that into smaller or several subplots, parts of the whole plot, and I'm going to then separate these whole plots by an area large enough that drift isn't going to be a problem. Okay, now what I can do is I can randomize the position of the corn, the potatoes, and the squash in the whole plot, and then I can apply a herbicide to all three of those crops in the whole plot, and it's far enough away from the next whole plot that drift isn't going to be a problem. Okay, and so they called this a split plot. So you would set up whole plots physically separate from each other and then split those into subplots where you'd vary the crops. Okay, so that's where the term split plot comes from, and where the terms that are still used of whole plot and subplot. Okay, so split plots have two types of factors, and so hard to change factors. These are applied to the whole plots. So in older literature, you'll often see factors referred to as whole plot factors, and these are what are in newer literature are often referred to as hard to change factors. Then the easy to change factors are applied to the subplots. So again, in the literature, you'll see both of those terms when they talk about subplot factors. Those are the easy to change factors. So those are used interchangeably, whether it's a hard to change factor, that's a whole plot factor, or an easy to change factor, that's a subplot factor. Okay, we restrict the randomization on the hard to change factors. Okay, so if you go back to our little picture, what we did was we said, okay, we're going to put herbicide B on all three crops. Okay, so we're restricting the randomization there. We're only changing the herbicide once between each of the three whole plots, whereas we're varying the corn, the squash, and the potatoes on all three whole plots. They get varied a lot more. Okay, so split plots naturally arise in many DOE studies. It used to be, more so than it is now, that building and analyzing a split plot was tricky. Okay, now we have good DOE packages that'll handle all of that for us. And split plots are implemented in Design Expert, and they're very easy to use. Okay, so that's a little bit of the history and background on split plots. So now let's just look at a couple examples and see how this is used. Okay, so I'm going to look at an example where what we want to do is DNA amplification. Okay, so we're going to look at this as a factorial design. So we're going to have 9 factors, and each of those 9 factors is going to have 2 levels. Okay, 3 of the factors that we're going to study are associated with the thermal cycler. So we're going to be doing our experiments on this 96-well plate. Okay, so that's what we're going to be doing our experiments on. But when we thermal cycle it, we have to pick the plate up and put it in the thermal cycler. Okay. So 3 of the factors are factors associated with the thermal cycler. The other 6 factors are associated with the sample preparation using an automated liquid handling system. So on here, we're going to be pipetting different levels of these 6 factors into individual wells. Okay, so the individual wells on the 96-well plate is our subplot. So the plate in this case is our whole plot, and the wells are our subplots. So we have one plate that has 96 subplots. So the key points here are each combination of the three thermal cycle factors are going to give us a 2 to the third or 8 combinations. Okay, so that means we have to use 8 different plates, because remember, when we use the thermal cycler, we have to pick up the plate and put it in there and run it through the thermal cycle. Okay, we're going to use a 2 to the 9 minus 1. So that's going to give us 256 runs. And those are going to be on 8 different plates. Okay, so our 9 factors are going to be 3 thermal cycle factors and 6 sample preparation factors. And if you take 256 and you divide it by 8, you're going to get 32. So there's going to be 32 subplots, or 32 of the cells are going to be used on that 96 well plates for our experimental units. Okay, our 3 thermal cycle factors are hard to change factors. So those are varied between the plates. So using the whole plot terminology, they could be called whole plot factors. In this special case where we're using a 96 well plate, those could be called a whole plate factor, because a whole plate sees the oven, the thermal cycler, when we put it in. Then the 6 preparation factors are easy to change factors, and they're going to be varied as at the subplot level. And so each one of these whole plots has 96 subplots, and we're going to use some portion of those. In this case, 32 for the experimental units and some additional wells for control units. Okay, so our 3 thermal cycle factors are the annealing temperature in the thermal cycler. It has 2 levels. The denatured temperature, and that has 2 levels. And then the denatured time, that has 2 levels. So that's going to be our 2 to the third, and define the conditions in the thermal cycler that we're going to cycle the 8 plates through. Now, our subplot factors are these 6 factors, and they each have 2 levels. Okay. And those are going to define the conditions for the cells within the plates. Those are our subplot factors. Okay, so we're going to have 8 combinations of the thermal cycle factors, and that's just laid out here. That's a full factorial 2 to the third. Now, on each one of those, we're going to put a plate in there and run it under those conditions, and that plate's going to have 32 units for the subplot factors. Okay, if we wanted to completely randomize this experiment, what we'd have to do is we'd have to do a single subplot unit on a plate, and also the measurement we're making is always relative to a control, so we always have to have a QC well. So we do a single unit on a plate, pick that plate up, put it in the thermal cycler. So now, instead of using 8 plates, you'd have to do the 256 experiments, each experiment on a separate plate. And this would amount to just having 1 cell on each plate, and it'd be a really big waste of resources. So what we're going to do is we're going to use a split plot to minimize the number of plate changes. Okay, so in the split plot, we're going to have 32 cells we're going to use. Those are the dark cells, and then we're going to have some QC cells. Okay, those are the gold cells on these plates. Okay, so we're going to have the 8 plates that are defined by the thermal cycle factors of anneal temperature, denatured temperature, and denatured time. And in those, we're going to vary the 6 reagent factors. Okay, and within the cells, 32 in each one, we pick it up and we run it through. And then we do the next combination of thermal cycle factors and subplot factors and pick it up and run it through, and so on. So with only 8 whole plot combinations, or plates in this case, we can get our 256 runs. And we're going to have 32 cells per plate, as opposed to 1 doing completely randomized. So this is going to be 32 times faster than if we did a completely randomized experiment. And these experiments are expensive. The equipment is expensive. It's hard to get time. And so it would be really impractical to do a completely randomized experiment. Okay, so let me show you how you do this using Design Expert software. I'm just going to switch to Design Expert. And to save time during the presentation, I'm not going to do all the typing. I'm going to read in just a little dummy file that has the names of the factors and their levels, so I don't have to type that in. Okay, but otherwise, you'd be starting with a new design. I'm just going to tell it to reuse the names of the factors. Okay, and I'd go down here to split plot, and I'd say I want to do a regular 2-level factorial. I've got 9 factors, so you'd pick 9 from the list. I've got 3 hard to change factors, so you can pick how many hard to change factors you want. Now, how many plates do we want to use? So that's groups. So groups are the same as whole plots. So I've got 8 groups I'm going to do, or 8 whole plots. How many runs do I want to do? Well, I want to do 32, not 16. So I'm going to do 32 in each of those 8 groups. Do 1 replicate. Okay, that's going to be a 2 to the 9 minus 1, or a half fraction. That's going to be a resolution 9 design. So that just means that main effects get aliased with 9-factor interactions. And so we don't have to be very concerned about aliasing in this. Okay, so then we'd say, okay, that's what we want. And this is just showing that the aliasing is not important. There's nothing of main effects with other main effects, or 2 factors with other 2 factors. Main effects are aliased with 8-factor interactions, and 2 factors are aliased with 7-factor interactions, so that's not going to be a problem. Okay, so we go to the next. Now this is why I read a file in, just to save this. The first 3 factors is where you'd put your hard to change. Remember we said there's 3 hard to change, so those are going to be the first 3 factors you put in. So that's our factors associated with thermal cycler. And then the next 6 factors are factors are going to vary within the whole plots, or vary at the subplot level, or in this case vary at the individual cell level on the plate. Those are the 6 factors. We're going to vary there and their levels. Okay, we're going to measure amplification, really how much more DNA do we generate. We want to generate as much as we can. What we're interested in here is signal to noise of 3 to 4. Okay, so the signal we want is, if you want to think of it as 3/4 of a standard deviation is what we're interested in, or the signal to noise is 0.75. When we go here, now we see the major cost of running a split plot. What do you see when you look at the power? Well, we have 2 groupings of power. We've got a group that the power is quite low, and then a group where the power is really high. What is that? Well, how many times did we change the whole plot factors? Well, there's only 8 plates, so that gets the power sort of if it's a 2 to the 3rd experiment. Okay. How many times did we change the subplot factor? Well, actually 256 times. So that gets the power of a 2 to the 9 minus 1 factorial. Okay. If you looked at comparing it to a completely randomized design, so if you did 9 factors as a completely randomized design, what you'd find is the power is somewhere close to what it was for the subplot factors, a little bit less. Okay, but much, much higher for the whole plot factors. So completely randomized, they all have the same power. Okay. The reason the power is low for the whole plot factors, as we talked about it, the whole plots are really a 2 to the 3rd experiment. The reason the power for the subplot factors is a little higher is as we go into the analysis, we'll see that there's 2 different error terms that get used. You get an error term for the whole plot factors, and that includes both the error associated with the whole plot factors and the subplot factors. Then we get an error term that we use for the subplot factors. That doesn't include the error for the whole plot factors. So that error actually gets reduced, and so you get a little bit higher power. So here we're starting to see the pros and the cons. The big con that we get here is the loss of power of the whole plot factor. The big pro is we can do the experiment, right? We can't do the completely randomized experiment. At least we can do the experiment. Now, a little pro is you also improve the power for the subplot factors. Okay, so that's also a little pro. But the big pro is we can do the experiments. Okay, so then we can finish this up, builds our design. And you can see there's 32 runs in group 1, so that'd be plate 1. Okay, and that's one combination. Notice the combination for the whole plot factors or the denature factors don't change. They're constant in group 1, and we're varying the subplot factor. Okay, then we've got the next group and the whole plot factors and the subplot factors. Now we've got this reminder, factor settings should be reset between groups even if the levels are the same. Okay, so if we look at this, the annealing temperature changes, so that we have to change. The denature temperature changes, that we have to change. The denature time is the same. Now, remember when we run an experiment, we don't really control any of the factor levels. We control the set points of the factor levels. And every time we move away from that set point and move back, there's some variation in how close we got to that target value. So what you need to do when you're doing a split plot experiment is if that factor doesn't change, you should still move the knob away and move it back so that you capture that normal variation around the set point. Now you should always do that in experiments, but in a completely randomized experiment where we're changing everything every time, if you don't do it for the factors, they still get changed a lot. It's not a big deal. In a split plot, since we're restricting the randomization, we're not changing the factors very much at all. In this case, we're only doing 8 sets of the factors. If you didn't reset that factor in between groups, you'd miss capturing the right variation for the whole plot factors. We wouldn't be able to separate out the variation due to whole plot and subplots to get the right error terms to do the tests. Okay, so that's all that's telling you is you have to remember and reset those between groups. Okay, so that's our design. I'm gonna get some data. For this by running a little simulation. But first I'm gonna go back to the PowerPoint, make sure I didn't skip anything. Okay, so I opened up our starter design, I built the design, we had the hard to change factors, the easy to change factors, 2 levels for each. We looked at the delta and the sigma. We saw the cost associated with doing the split plot, that we lose power for the whole plot factors because they aren't varied very often. The big advantage is we took an experiment that was impractical and we made it doable so we can do the experiments. And then a little side benefit also is we improve the power associated with the subplot factors. By separating the error term into whole plot and subplot, we get a little less error associated with the subplot factors. Okay. So the split plot design is really two designs, the whole plot factors for setting up the thermal cycler and the subplot factors for creating the samples in the wells or cells in each one of the plates. Since the well-to-well subplot variation occurs only within a plate, the plate-to-plate variation is not included in that. That's where we get that separation error and get the little higher power for the easy to change factors or the subplot factors. The error associated with resetting the thermal cyclers plate to plate is included in the whole plot variance. That gets the total error from both. Other considerations, every run varies due to the resetting of the factor levels. Whole plot factor treatments only change by groups. Subplot treatments may be altered on every run to account for the different errors we have to use. Now this is a two-level factorial. If we want to use half normal plots or really any other tool, we have to account for the fact that they're from two distributions. The whole plot factors are from a normal distribution with larger error, and the subplot factors are from a normal distribution with a smaller error. So we'll have to take that into account when we do the analysis, just like we took that into account when we did the power calculations. Okay, so let's go through and see how to analyze this. Let me switch back to Design Expert. Okay, so I put the analysis in there. So we go down to amplification. Okay, method we're going to use is restricted maximum likelihood. Okay, that's our only choice for a split plot. And what that refers to is this fact that we're going to try and divide the error up into two groups. And we're going to do that in the way that makes the most sense based on the data. And that's what maximum likelihood means. It's what makes the most sense based on the data we have. Okay, so I'm going to start the analysis. You know, this looks pretty much like any other factorial, except I've got two different plots, one for the subplots, one for the whole plots. Okay, now when I go in here and I select the factors that look significant, you can see I selected D. Okay, that's a subplot factor, AD. Okay, now what we did in Design Expert as a matter of convention, since we've always used capital letters to represent the factors, we wanted to differentiate the whole plot factors. So we're using letters, but we're using the small uncapitalized version of the letters. So AD tells me that's a whole plot by subplot interaction. So what's nice is, remember I said the whole plot factors is like a 2 to the third. It's got 8 experiments. Well, the only factors that get that error are the whole plot factors, so A, B, and C, and their interactions, AB, AC, BC, ABC. So any of the whole plot by subplot factor interactions get the subplot errors. So they get the smaller error. So it's really just like a 2 to the third. Just the factors associated with that 2 to the third are the whole plot factors. Any of the interactions with subplot factors get that smaller error. And so if the factors that you're studying whole plot, even though they have low power, if they interact with the easy to change factors, you're going to be able to pick those interactions up, and then you can put the hard to change factors in to maintain the hierarchy or heredity of the model. Okay, so we've got D, AD, FJ, J, and F. So when I click on whole plots, it's saying we're not maintaining hierarchy. Do you want to do it? If we say yes, then we'll see that it automatically selected A. So okay, you had AD, so I'm going to put A in. Now it looks like there's another factor here, B. So the annealing time and the denatured temperature seem to be the drivers here. And then we can just go to Anova. Again, this is using a restricted maximum likelihood. and we get a kind of like a regular Anova, but it's got two sections because remember the whole plot terms get one error term and get a certain degrees of freedom, and the subplot terms or the easy to change factors have a different error term and obviously a different number of degrees of freedom because they're varied a lot more often. Now the REML part of this, so the restricted maximum likelihood, what it's doing is taking the total variation in the design and trying to divide that into what's due to groups and what's due to the subplots. And you can see the standard errors there. And that's the random part, is how do we figure out how to put those errors? And that's the part that uses a restricted maximum likelihood. Okay. Otherwise, this Anova, you can look at it like normal. And we've got kind of normal statistics, and we've got our equation. Okay. We can go to diagnostics. We've got the normal plot like we'd normally have. We've got the studentized residuals versus the externally studentized residuals versus predicted, the residuals versus run, the residuals versus each one of the factors. We've got the box cock plot. We got the predicted versus actual. We've got the Cook's distance. Now, remember there's this part where we're trying to estimate the factor effects. That's what all this is related to. We also tried to estimate the error terms. And so this is the effect of the runs on the fixed effects, or that's the effect on these model terms. Okay, but now remember we also estimated these variance components. So if you go into Cook's distance, you can select a graph and you can say, okay, that's the part, the fixed equation. What about how did these runs affect the variance components? Okay, and so that tells you that. So you can see there's so many runs, none of them have much of an influence. Okay, but we can check that as well. And we got leverage, that's kind of the normal thing. DF fits, you could go through and do that for each one of the factors. Now we've got a new graph here, which again is how do the runs affect the variance part. We can do the covariance, either the ratio or the trace. Okay, so again, it's giving us diagnostics on not only how do the runs affect the equation, which is what we normally look at in a completely randomized design, but also got 3 extra graphs that tell us a little bit about how the runs affect the estimate of those variance components. Then we get to the model graphs, and those would be used as normal. I'm going to kind of skip that, not because it's not important. It is important, but I have limited time here, and I'm not trying to teach you about analysis. And in general, I'm trying to teach you about split plots. So I'll just jump right to the optimization, and we could run the optimization and figure out, okay, set this factor at its low level, denature temper at its high level. Denature time didn't have an effect. It wasn't in the model, so it's set at the midpoint. Primer low, didn't have an effect. Probe low, didn't have an effect, didn't have an effect. And the gold low, and that's going to get our amplification up to about 15.9. That's a quick example of how to set up and analyze a split plot as a factorial. Now let's look at doing a similar process, but with a different design. And so I wanted to do a response surface design, but I also wanted to do a mixture design. So I decided to just do them both. Remember, mixture is just a special case of a response surface. And when you study mixtures and process factors, it seems like it's almost most of the time that one or the other is really hard to change, and you have to do a split plot. So this is a pretty common case. So in split plots, when you're doing combined mixture process, it may be that the process factors are really hard to change. So you could think of, I'm going to run a design. So in this case, I just showed 2 process factors and a 2 to the 2. I could have showed them as a central composite design or a 3-level design or an optimal design, it doesn't matter, but they're the ones that are hard to change. And so for each combination of process factors, you're going to do a number of mixture experiments before you change to the next level of the process factor. And you do a number of mixture experiments before you change to the next level and so on. So that's when the process factors are hard to change. When the mixture factors are hard to change, the same thing, you have any kind of design, but you're going to set up a particular mixture combination, then you're going to go through several combinations of the easy to change process factors, then you go to a different mixture combination, go through several different experiments with the process factors, then you go to a different one, mixture, do the process factors. Okay, so either the process factors or the mixture factors could be the hard to change factors. Okay, we're going to look at a case of where we're going to look at reverse phase HPLC. So HPLC is high performance liquid chromatography. For those of you that are really old, like me, it used to be called high pressure liquid chromatography. It's the same thing. The form of column chromatography used in biochemistry and analytical chemistry to separate and identify components based on their polarities and interactions with the column stationary phase. And reverse phase just means that we're going to have a non-polar stationary phase and an aqueous or moderately polar mobile phase. So regular HPLC, those are just reversed. And there's a little picture of a HPLC. So very simply, excuse me, you got a mobile phase, that's our mixture. We're gonna mix up different things in the mobile phase. That's delivered to an injector where we inject the sample, and that then is the carrier of the sample through the column, the HPLC column. That comes out and goes to detectors, and we measure the different peaks. Okay, now this is more done digitally now, but basically we're trying to see how to separate out different components in that sample based on how it's held up going through the column. Okay, so our mobile phase is going to be the easy to change factors in this case. It's easy to mix up these things. So we're going to vary water, methanol, and acetonitrile. Okay, and the tetrahydrofuran is in there, but that's going to be fixed. Okay, so we're going to have 3 factors we're going to vary and put in a fixed amount of this, always 5%. Okay, and on the column, we're going to vary the particle size. So this is columns packed with a bunch of little different, packed with a bunch of little spheres. So it actually is a column packed with spheres. And we're going to look at using spheres of different diameters, 3.5, 6, or 10 millimeters. And then these spheres have pores in them. You can vary the pore size. So each one of these is going to have 3 different pore sizes, okay, 100, 200, or 300. Now there's other column factors we could vary, but the bonded phase is going to be fixed, and the column dimensions are going to be fixed. So we're not physically going to change the column, we're just going to change what it's packed with. Okay, and so that's going to be our hard to change, because to change this, you have to take the column apart, throw away what's in there, clean it, repack it. And so to do a completely randomized experiment would be very time consuming and very, very expensive. Okay, so the mobile phase is easy to mix and change. The column factors, the particle and pore sizes are difficult and time consuming because we got to physically break down the equipment, take the column out, unpack it, clean it, repack it. The experiments will be easier, faster, and less costly if the mixture components are specified as easy to change and the columns are specified as hard to change. Okay, so we're going to be in here, we're going to be running a number of different mixtures for each combination of the process factors. Okay, let's see how to set this up. So again, I'm gonna use a dummy starter file to save the time of typing in all the factors. Okay, so we start here and we go to custom design. We're going to do an optimal design. Okay, we're going to have 4 mixture components. Remember, one of those are going to be constant, 3 of them are going to vary, but there's 4 components we're going to put in, and 2 processor or numeric factors. Okay, this is going to add up to 100%. We're going to use volume percents. We're going to vary the water, the methanol, and the acetonitrile between these limits. We're always going to put in 5% of the last ingredients. That's a constant. Okay, so basically these remaining ingredients will make up 95%, and we'll add 5% to get 100. And these are the easy to change factors. Okay, we could have made those the hard to change. Now here, what I did was I changed from, by default, this would have been continuous. Okay, but we changed it to be discrete. And I think this was, what was this? So it's 6. Okay, now the reason we changed these to be discrete is you order these particles with a certain diameter and a certain pore size, and you can only get what's in the catalog. Okay, so we could buy particles that have a diameter of 3.5 and a pore size of 100, 200, and 300, particles that are diameter of 6 with 100, 200, 300 pore size, and so on. Okay, so we can only run the ones we can actually obtain. Okay, then it sets up our design, what we're going to do. Okay, so we're going to do a quadratic by quadratic. It requires 6 groups. You want to have some, it's telling you need 9. So well, we're going to go in here and say to be able to do the Remel, you got to have some extra groups. So we're going to do 3 additional groups so we get our 9 that we need. We're going to put in a couple extra model points just because that's what the design that I took this from had. So we're going to have a total of 45 runs in 9 groups. Okay, so how many runs are going to be in each group? Well, 45 divided by 9 is 5. So we're going to set up the column, and then we're going to do samples using 5 different combinations of the solvent, different mixtures of the solvent or the carrier, the mobile phase. Okay, what we're going to measure is the height of an equivalent theoretical plate in millimeters. And the smaller you can make that, the smaller you can make the height of an equivalent theoretical plate, the more plates you can get into the column and the better separation you get. So we want to minimize that number, make the height of a equivalent theoretical plate as small as possible so we can get the best separation out of the column. Okay, so that's going to build our design. It takes a little less than a minute. And while that's building it, I'm going to switch back here and make sure I didn't skip anything. 4, 2, easy, hard, discrete. 6, 9, 45. Okay, so if we look at the design, we can see we've got one combination of the process factors, so that's factors 5 and 6. Then we do 5 combinations of the mobile phase. Okay, the 3 varying and the 1 is a constant. Okay, then we change the factors. for the column, and we do 5 more. We change the column, do 5 more, and so on. Okay, and again, it's reminding us if there's a case like here where the factors don't change, we'd still reset it. Here, that's not really a problem because we're actually repacking the column each time, so you have to change the factors. Okay. Now again, I'll simulate results so we can see how to do the analysis. Go in here. Again, we're going to use restricted maximum likelihood. Okay, now we get something that looks more like what we get when we have a response surface, a mixture, or a combined design where we're fitting a polynomial model. The default is a quadratic cross with a quadratic. If we went ahead and fit that, we'd see that there's a lot of terms that don't look like they're significant. So we'd probably want to reduce this model. And you do this just like you would any other model. I like to use AICC and I like to use backward, so that's what I'll use. You could use another method or several methods and compare the models. In this case, they all come out pretty much the same. Okay, so we accept this. We say yes to hierarchy. Now you get a much smaller model. Where the terms are significant for the most part. There's a couple terms that aren't significant, and we can see those are terms A, F, B, E, and C, E that were added to maintain hierarchy. Okay, so that's our fixed model. Again, we can see the rebel part where it took the total variation and divided it up. Okay, to give us the components. Got our statistics that we look at. We've got our model. We can go to diagnostics. And again, we've got the diagnostics that are associated with the fixed portion of the model. Okay, and the first 3 boxes, and we get to Cook's distance and Cook's distance for the fixed portion of the model. And we can also do Cook's distance associated with the random part, or again, the random part is estimating those 2 variances, dividing up the variance between the easy to change and the hard to change factors. Okay. Leverage, DF fits, and then we get, again, covariance for the ratio and the trace. We can go to the model graphs, and we can look at the model graphs for the mixture part and see how that changes as we change the process factors, or we could switch it. We could look at the process part and see how that changes as we change the mobile phase. Again, to save time, I'm going to jump to the numeric optimization. Remember, we wanted to minimize this. Just put in a number a little bit smaller than what we got. Otherwise, I'll get a desirability of 1. I only know that because I already ran it before, in preparation for this talk. We can look for solutions, and we find the level of water we want to use, methanol, acetonitrile, particle size, pore size. Oh, but remember, we can't necessarily get particle size and pore size to be whatever I want. Okay, so if I go back here, notice these have a little hashtag in front of them. That's because those are discrete factors, and I can restrict my optimization to only pick levels of the factors that I studied, only pick the discrete levels of the factors. And I can rerun this. Okay, now it's going to find levels of the mobile phase, okay, but it's going to restrict the column factors to the actual particle diameter and pore size that I can purchase. Okay, so after crossing, you get kind of a weird result when you're hard to change factors, the process factor, and you cross it with the mixture factor. Because you're crossing with the mixture factor, everything's easy to change by hard to change. There's no hard to change factors that appear on their own. So everything gets the smaller error of the subplot factors, which is kind of nice. It only happens in a combined design. Most of the experimental error is changing the process factor, so you can see the group variance is the bigger portion of the total variance than the residual. We went in and minimized the height of a theoretical plate so we can get more plates into our column to get better separation. And so that's just an example of doing a response surface. So hopefully what you observed with these examples is setting up the design's different as a split plot. The computer has to do more work to figure out the variance components. You know, that's that RML part, restricted maximum likelihood, just figures out how to divide the variance that makes the most sense for the data you have. Maximum likelihood makes the most sense. Okay. You don't really have to worry about that. The computer does that. So setting it up and doing that, you do have to be aware of the pros and the cons, which I'm going to go through in the summary. But the computer takes the work out of it. Once you've got it analyzing it, it's pretty much the same as a regular factorial, except you got to pick the two factors separately. You should look at those plots to make sure that none of the runs are throwing the variance estimates out of plots, so the Hooke's distance and the covariance plots. See the effect of the runs on the individual runs on the variance estimates. But it's pretty straightforward, pretty easy to do. It's the same as a regular factorial for the most part, and the same as a response surface or a combined mixture process or a mixture if you're just doing a mixture. Okay, so let's summarize. So the pros are, the big pro is it's practical. I mean, if your hard to change factors really aren't hard to change, you're better off doing a completely randomized design. It's only when restricting the randomization really makes the design practical. Now I've seen a couple, well, this is in like 40 years experience. So seeing a couple designs where people have had hard to change factors because they're really interested in a subset of the factors, the subplot factors, and they want to reduce the error because some of the factors contribute a majority of the error, so they make them hard to change factors. And they do that more to set the error aside than to estimate the factor effects, so they can get better estimates of the subplot. That's really, really rare. Okay. The main reason is the hard to change factors are really hard to change, and we can't practically do the experiments if we do a completely randomized experiment. So you have to consider the cost savings of the split plot versus the loss of power to detect the hard to change factors. That's the main consideration. These are flexible. Factors that have large experimental units can be easily combined with factors that have small experimental units. That's why I wanted to use the DNA amplification, because that's a really good example where the plates are large units, right? And they have a lot of small units, the cells, 96 of them, or 384 or 1,500 and whatever units on each one of these large units. And a split plot gives me an easy way to combine those large units and small units, code for that in the analysis. And they're more powerful tests for the subplot effects. Well, I guess I mentioned that up here. Sometimes you make things hard to change, not because they're hard to change, but because they have a lot of variance, and you really want to set their error aside so you can get better estimates of the things you're calling easy to change factors. Excuse me. Cons, they're unfamiliar, so they result in different errors for the whole plot versus the subplots. We need to use this restricted maximum likelihood to find the error components. If you were doing this on your own, this would be a major hurdle. And if you went ahead and said, I'm going to ignore that, I'm going to apply a standard Anova, what happens is you're underestimating the error for the whole plots, and you're likely to affect too many hard to change factors, and you're overestimating the error for the subplots, and you're likely to miss significant easy to change factors. Okay. Luckily, modern computer DOE packages will handle this for us, so there's really, that first bullet point isn't much of an issue, other than you're aware of it. And when we want to restrict the randomization, you use the split plot rather than just sorting and ignoring and treating it as a completely randomized experiment. Actually set it up as a split plot and analyze it that way. The big con is you lose power on the hard to change factors. They have a larger variance and fewer changes to overcome that larger error. Remember, you do get an advantage that the interactions between hard to change and easy to change have the error of the easy to change. So if the factors interact, you're going to catch it anyhow. Hard to change and easy to change factors are tested against different noise. This can result in hard to change factors being not being statistically significant, even though they're large, right? The power is real low, so they may have large effects. You have to watch for that, and if it's a big effect, you have to be concerned about that. And you may find, in some cases, the easy to change factors have really small errors, if most of the error is associated with the hard to change, and you'll find things that are statistically significant, even though they're so small, they're not practically important. You just have to be hyper aware of what you should always be doing, which is looking at the size of the effect and saying, if that was real, would it make any difference? Okay, you should always do that, whether it's a split plot or not. But here you have to be extra careful because it's easy to miss big effects in the hard to change, and it's easy to find significant but not important effects in the easy to change. Things to remember: not randomizing these groups increases the risk of getting a lurking factor, right? If something was changing over time, since we're not randomizing the hard to change factors, it's more likely that that lurking factor can get confused with hard to change factors. So it's really important when you do in a DOE, but particularly when you do a split plot, to try and hold everything you're not studying constant. When group changes occur, the factors that are not changing should be reset. We talked about that. Factors not reset, then that creates another restriction on the randomization that's not accounted for in the analysis. And so you really want to make sure you reset them. And so that's my presentation. In a day or two, this will be posted on the website if you want to look at it. Hopefully, Sherry has been answering your questions. If you've been asking questions, I haven't looked, so I don't know. If you have additional questions, now that this is over, you can send an email to stathelp@statease.com. We do have some training, online training that you might be interested in. If you want to know more about that, we have a modern DOE for process optimization. We have a course on mixture experiments for optimal formulas. We have industry-focused DOE classes on pharma and food and other areas. And so we have classes you can take online as individuals or as groups. We have a lot of different options. So you can take a look at those on our website. And if you want more information, you can contact workshops@statease.com. So I want to thank you for attending my presentation. I hope it was time well spent for you, and I enjoyed doing it. And with that, I'm going to just end the presentation. Thank you.